Dear students, in this lesson we are going to talk about digestion. Digestion is the breakdown of large, complex organic molecules into smaller components that can be used by the body. Molecules need to be small enough to diffuse across plasma membranes. There are two main types of digestion. The first one is mechanical digestion, and the second one is chemical digestion. The mechanical digestion, it starts in the mouth. Physically breaks down the food in the mouth by chewing. Smaller pieces increases the surface area for digestion, further digestion rather. This also happens in the stomach. The churning of food by the muscular stomach walls breaks down the food into further pieces. The second method of digestion was chemical digestion. The enzymes are used to chemically break down the complex food substances into their simplest forms in order to be able to be absorbed into the bloodstream. Example, starch is turned into its simpler form, maltose, by the action of the amylase enzyme. The chemical digestion of certain molecules take place in specific areas of the gastrointestinal system. For example, the starch, or generally saying the carbohydrate digestion, occurs mainly in the mouth and small intestine. Protein digestion occurs mainly in the stomach and the small intestine. And fat digestion occurs mainly in the small intestine. So why must the food be digested? Large molecules of food are unable to pass through cell membranes, thus must be broken down into smaller molecules so that they can diffuse through cell membranes into the bloodstream. And by the bloodstream, as we all know, they are carried to the intracellular space for the benefit of each cell of our body. And we have steps of digestion, which can be written as ingestion, digestion, absorption, assimilation, and egestion. Ingestion is the consumption of taking in of nutrients, and it is our mouth for the entry of the food and where ingestion is made. Digestion is the chemical breakdown of large organic molecules into smaller components by enzymes. This is the absorption is the transport or delivery of digested nutrients to body tissues. And assimilation, this is the conversion of absorbed food into the substance of the body for the benefit of our body. It's the use of the absorbed food. And finally, we have egestion. This is the elimination of food waste materials from the body. Let's start with the mouth. Ingestion is accomplished by the mouth. The teeth masticates food and increases its surface area for a better digestion. The saliva in mouth moisten and soften the food ingested. The saliva contains mainly water, mucus, and salivary amylase, the enzyme to break down carbohydrates. The pH of saliva is slightly acidic, average a 6.7 value. The starch in the ingested food is converted to maltose with the salivary amylase. The tongue, it's a very powerful organ, mixes the food with saliva and rolls it into a bolus before swallowing. If we look at the esophagus, it is the hollow muscular tube connecting the pharynx and the stomach. It is approximately 25 centimeters long. It functions include to secrete mucus and basically the movement of food from the throat to the stomach using the muscle movement called peristalsis. Peristalsis is the wavy movement of the esophagus to carry the food to the stomach from the mouth. If somehow the acid in the stomach refluxes into the esophagus, it's felt like a heartburn. Now, if we look at the sketch here, we can see that once the food is ingested and chewed and brought to a bolus in the mouth, it is pushed by the tongue towards the pharynx, and as the food reaches to the laryngopharynx area, laryngeal pharynx, the epiglottis pushes toward the tracheal entry, closing the trachea and giving way to the passage of the food into the esophagus, where this epiglottical closure prevents the food getting into the trachea. And once the food is pushed past the laryngopharyngeal area to the farther parts of the esophagus, then the larynx reopens for inspiration. If we take a look at the stomach here, it's a J-shaped muscular bag that stores the food you eat, breaks it down into tiny pieces, mixes food with digestive juices that contain enzymes to break down proteins and lipids. Acid in the stomach kills bacteria ingested together with the food. Food found in the stomach is called chyme. The functions of the stomach can be expressed as it stores the food ingested for some time. It produces the intrinsic factor, IF, which is important for the absorption of vitamin B12 at the small intestine. It digests proteins by denaturation of the proteins. It controls the amount of food released to the small intestine. Produces the hormone gastrin, which helps the production of pepsin and hydrochloric acid in the stomach and bile in the liver. It buffers the food condition if it is too alkaline. If we move on to the small intestine, the small intestines are roughly 7 meters long. Lining of intestine walls has finger-like projections called the villi. The singular of villi, singular of is villus. And as you can see in this picture here, these are the villi of the intestinal wall, which increases the surface area of the intestines for a better absorption. These villi are covered with microvilli, which further increases the surface area for a better absorption of the ingested food. The majority of chemical digestion occurs in the first three sections of the small intestine. And the first part of the small intestine is known as the duodenum. This section also contains an opening from the bile duct and pancreatic duct through which bile and pancreatic enzymes enter the small intestine. Food enters the small intestine as a semi-solid mixture known as chyme, and it comes from the stomach. The chyme is acidic due to the hydrochloric acid in the stomach, so it needs to be neutralized. The presence of chyme in the small intestine triggers the conversion of prosecretin into secretin, which is absorbed into the bloodstream and carried to the pancreas. Secretin causes an increase in pancreatic and bicarbonate secretion and a decrease in gastric hydrogen secretion or acid secretion. Secretin stimulates the secretin of bicarbonate-rich pancreatic fluid and helps the digestion and alkalinization of the food. And after passing the small intestine, the food passes into the colon, which is the large intestine. The colon is about 1.5 meters long, 1.5 meters long. It accepts what small intestines don't absorb. The first part of the colon starts at the bottom where there is an appendix at the beginning of the colon. The small intestine connects to the colon, and the first part is the ascending colon, which goes outward to the to backwards of the liver in the body. The second part runs from the right side of the body towards the left side of the body, and it is called the transverse colon. And the last part of the colon is named as the descending colon. Ascending colon reabsorbs the excess water and nutrients that could not be digested in the small intestine. The transverse colon connects the ascending colon with the descending colon. It also helps the absorption of water and some electrolytes. Finally, helps the formation of feces to pass it to the descending colon. And the descending colon pushes the feces downward to be eliminated. It connects with a formation called the rectum. The final part of the descending colon is the rectum. It is the end part of the colon. It's a short-term storage which holds feces before it is expelled. From here, the stool passes to the anus and it is discharged. The anus. The human anus is the external opening of the rectum, and it is the area where digestion is accomplished. It starts with ingestion in the mouth and it is completed with egestion from the anus. Its closure is controlled by sphincter muscles. Feces are expelled from the body through the anus during the act of defecation, the primary function of the anus. Any diseases that affects the defecation, either the rectal or anal level, gives the rise to the containment of the stool, leading to a state called megacolon. And in our digestive system, we have our accessory organs for digestion. And these accessory organs are the salivary glands, the liver, the gallbladder, this green structure here, and we have the pancreas. These are the accessory digestive system organs. The salivary glands, we have three main salivary glands in our face. The parotid glands, are the biggest ones, the submaxillary glands and sublingual glands. These are responsible for producing saliva into the mouth for the initial digestion of the ingested food. The liver is a large accessory organ of the digestive system that is constantly producing a fluid known as bile, and bile is stored in the gallbladder, and it is discharged whenever it is needed into the first part, the duodenum part of the small intestine. The presence of lipids in the small intestine trigger the release of the hormone cholecystokinin, which triggers the release of bile from the gallbladder. Bile contains bile salts that emulsifies fats, which means it breaks them into smaller droplets so they can be digested. The gallbladder itself stores the bile in between the meals, secretes bile into the duodenum during the meals via the bile duct. Bile contains bile salts, pigments, and cholesterol and phospholipids. Bile is an emulsifier. It is not an enzyme, and it dissolves fat into the watery contents of the intestine. If we look at the functions of the liver, the working cells of the liver are known as hepatocytes. Hepatocytes have a unique capacity to reproduce in response to liver injuries, and the liver is one of the organs one of the first organs, maybe we should say, in the body to be bombarded with the detrimental hazardous material, and it needs to be very capable of regenerating completely. Liver regeneration can occur even after surgical removal of a portion of the liver or after injuries that destroy parts of the liver. However, despite this capacity of the liver to react to a damage and repair itself is remarkable, repetitive insults can produce liver failure and ultimately death. The liver is a metabolically active organ responsible for many vital life functions. The primary functions of the liver are bile production and excretion of the bile, excretion of bilirubin, cholesterol, hormones and drugs, metabolism of fats, proteins and carbohydrates, enzyme activation, storage of glycogen, vitamins and minerals, synthesis of plasma proteins such as albumin and clotting factors, and blood detoxification and purification, regulation of blood glucose levels, iron storage, and deamination of amino acids, and there are some other minor functions as well. So liver is just like a factory in our body, doing lots of work and capable of regenerating itself under insults. Due to these important activities, the liver is exposed to a number of insults and is one of the body's organs most subject to injury. That's why it has a capacity of regenerating itself. If we move on to the pancreas, the pancreas is an accessory organ of the digestive system. It releases chemicals to aid in digestion. Secretin will stimulate the pancreas to release a solution containing bicarbonate ion into the small intestine, where it will neutralize the acidic chyme and raise the pH from 2.5, the pH of the chyme, from 2.5 to almost 9. This inactivates the pepsin. Well, the functions of the pancreas with the carbohydrates, that when we take a long chain carbohydrate, amylase enzymes are released from the pancreas that break large carbohydrate chains into smaller chains called disaccharides. Di means two, and each molecule of carbohydrate will contain two, each part of carbohydrates will contain two molecules by the breakdown of the long chains of the carbohydrates. Then the small intestine releases the disaccharide enzymes, which break down the disaccharides into monosaccharides, single molecular of sugars, so that they can be absorbed and used by the body. If we look at the two pancreas's functions with the proteins, we see that there is an enzyme called trypsinogen, a protein digesting enzyme, and it is released into the small intestine, where it converts into trypsin, and it breaks down large protein chains into smaller chains. When you intake a large protein chain, it is broken down into smaller chains. And this is called the process of denaturation of proteins. The final step in protein digestion occurs with the release of erepsins from the pancreas, and they break the smaller chains into individual amino acids, and it is the amino acids that are absorbed into our body for further use. Now let us look what happens with the digestion of carbohydrates in the small intestine. There are three carbohydrate products which are absorbed by the small intestine. Namely, there are the glucose, galactose, and fructose. The majority of carbohydrate digestion occurs in the small intestine. The main enzyme is pancreatic amylase, which yields disaccharides from starch by digesting the alpha-1,4 glycosidic bonds of the carbohydrates. The disaccharides produced, which are the maltose, maltotriose, and alpha-dextrins, they are converted to glucose by brush border enzymes. Disaccharides occurring naturally in food do not require amylase to break them down because they're disaccharides already, and they're converted into lactase, sucrase, and trihalase by border enzymes in the surface of the intestinal system. And these are hydrolyzed the compounds into molecules of glucose and galactose and fructose. If we look at the proteins, what's happening with the proteins? The protein digestion begins in the stomach with the action of pepsin, which breaks protein into amino acids and oligopeptides, and this event is called the denaturation of proteins. The process of digestion is completed in the small intestine with brush border and pancreatic enzymes. They split the oligopeptides into amino acids, dipeptides, and tripeptides. Di means two and tri means three, so there will be amino acids, dipeptides, peptides with two molecules, and tripeptides, peptides with three molecules. With this process, proteins become ready to be absorbed via the small intestine. And if we look at the lipids, lipids are hydrophobic and thus are poorly soluble in the aqueous environment of the digestive tract. Their digestion is started by lingual and gastric lipases, but this only digests 10% of ingested lipids. The remainder of the lipids are digested in the small intestine. Here, bile aids digestion by emulsifying the fat goblets into smaller chunks called micelles, which have a much larger surface area to be acted upon for breakdown. The pancreatic lipase, phospholipase, A2, and cholesterol ester hydrolase, three major enzymes involved in lipid digestion, hydrolyze the micelles, breaking them down into fatty acids, monoglycerides, cholesterol, and lysolecithin. With this process, lipids become ready to be absorbed into the bloodstream. And what about water absorption? Now, the average adult usually ingests 1 to 2 liters of water each day, but the fluid load of the small intestine is almost 9 to 10 liters, 8 to 9 liters being added by secretions of the gastrointestinal system. So you imagine the huge amount of secretions throughout the gastrointestinal system from the mouth until the anus. They produced secretions 8 to 9 liters per day. It's a great load and it's great loss of water from the body into the gastrointestinal system. Most absorption of water and electrolytes occurs in the small intestine, with some water absorbed in the colon as well. Water absorption is dependent on the absorption of solids such as sodium and chloride. Sodium is absorbed from the intestinal lumen. Most use the co-transport with glucose and amino acids and the sodium-hydrogen exchange, which allows sodium ions to move from the lumen into the enterocyte. Enterocyte is the name given to the cells of the intestine, enterocyte. Now, sodium is rapidly removed from the enterocyte via the sodium pumps, allow water via osmosis to follow either transcellularly or paracellularly, or namely between the tight junctions of the enterocytes. Water as well as sodium ions can then diffuse into the capillaries. Dear students, now we're moving on to another topic, which is called acid-base homeostasis. This is the balance for the metabolism of the human body. Acid-base homeostasis involves chemical and physiologic processes responsible for the maintenance of the acidity of body fluids at levels that allow optimal function of the whole individual. The chemical process represents the first line of defense to an acid or base load and include the extracellular and intracellular buffers. The physiologic processes modulate acid-base composition by changes in cellular metabolism and by adaptive responses in the excretion of volatile acids by the lungs and fixed acids by the kidneys. The need for the existence of multiple mechanisms involved in acid-base regulation stems from the critical importance of the hydrogen ion, H+ ion, which stands for hydrogen, concentration on the operation of many cellular enzymes and functions of vital organs, most prominently the brain and the heart. Metabolic pathways are continuously consuming or producing hydrogen ion. The daily load of waste products for excretion in the form of volatile and fixed acids is substantial. So what does pH stands for? pH stands for potential for hydrogen or power for hydrogen. From here we can understand that the basic element for the decision of pH value in the body is mainly hydrogen. If we look at the effects of pH, the most general effect of pH changes are on enzyme function. Also, they affect the excitability of nerve and muscle cells. As pH drops, excitability drops as well. And as pH increases towards the normal, excitability increases as well. The acid-base status of the human body must always be kept in a balance, otherwise life cannot be possible. The acid-base balance is primarily concerned with two ions, the hydrogen ions and bicarbonate ions. The arrangement of hydrogen and bicarbonate concentrations in body fluids are common in disease processes. Then is when the acid base is mostly affected. Hydrogen ion has special significance because of the narrow ranges that it must be maintained in order to be compatible with living systems. Maintenance of an acceptable pH range in the extracellular fluid is accomplished by three mechanisms. The chemical buffers, they react very rapidly, in less than a second. The respiratory regulation that reacts rapidly in seconds to minutes. And finally, the renal regulation, which reacts quite slowly from minutes to hours. So these three chemical, respiratory and renal regulation of acid-base regulation is very, very important. The chemical buffers. The body uses pH buffers in the blood to guard against sudden changes in acidity. A pH buffer works chemically to minimize changes in the pH of a solution. Respiratory regulation. Carbon dioxide is an important byproduct of metabolism and is constantly produced by cells. The blood carries carbon dioxide to the lungs, where it is exhaled. And remember, the blood is carried with the venous side of the vessels. So the carbon dioxide-rich blood is the venous blood. So the venous blood has a little bit less pH value when compared with the arterial blood, because there is less carbon dioxide in the arterial blood and more oxygen there. So when breathing is increased, the blood carbon dioxide level decreases and the blood becomes more base. When breathing is decreased, the blood carbon dioxide level increases and the blood becomes more acidic. This is called, the first one is called respiratory alkalosis, and the second one is called in the clinical applications respiratory acidosis. If breathing drops and carbon dioxide rises and the pH of the blood drops, this is called respiratory acidosis. So by adjusting the speed and depth of breathing, the respiratory control centers and lungs are able to regulate the blood pH minute by minute. If the degree of deterioration surpasses the degree of compensation rate of the lungs, then the body pH shifts to acid despite the lungs' efforts. And when we look at the kidney regulation, excess acid is excreted by the kidneys largely in the form of ammonia. The kidneys have some ability to alter the amount of acid or base that is excreted, but this generally takes several days. So enzymes, hormones, and ion distribution are all affected by hydrogen ion concentrations. Let's take a look at the acids. Acids can be defined as a proton donor, which is the hydrogen ion. In this case, the hydrogen ion itself is a proton. So if a substance is giving out a proton, if it is a proton donor, then that substance is most probably an acid. The hydrogen-containing substances which dissociate in solution to release hydrogen plus hydrogen ion are said to be acids. Many other substances, like carbohydrates, also contain hydrogen, but they are not classified as acids because the hydrogen is tightly bound within their molecular structure and it is never liberated as free hydrogen ion. So the liberation of free hydrogen ion is very important for a matter to be accepted as an acid. Physiologically important acids include in the body carbonic acid, H2CO3, phosphoric acid, H3PO4, pyruvic acid, C3H4O3, and lactic acid, C3H6O3. These acids are dissolved in the body fluids. Let's take a look at the bases. The bases are defined as proton acceptors. So if a solution is accepting protons, here in the context of body, the hydrogen, they are accepted as the bases. Molecules capable of accepting a hydrogen ion. So they are in the form of OH, oxygen, and hydrogen 1 negative. So when they take a positive hydrogen ion, they're ready to take a positive hydrogen ion, and they form the H2O, which is the water. Physiologically important bases include bicarbonate and biphosphate. Now here we have a scale of the acid base pH values, and here we can see that the battery, for example, is one of the truly known acidic material, and then we have the stomach acid. So we're carrying a hydrochloric acid in our stomach and the pH is here, and we have the citric acids like lemon, vinegar, and we have the tomato, coffee, milk, and water, which comes towards the neutral range from the battery to the water, and this part forms the neutral range. And then from the acid towards the neutral, we start to go to the basic state, alkaline state, which we have, where we have the baking soda, the stomach tablets that we use, like the Reni, the Talcete, etc., the ammonia solutions, the soap, the bleach, and then drain cleaners. So as you can see, drain cleaners, as to the contrary of the general thought, drain cleaners are strong alkalines, strong bases, instead of being acids. They're not acids. So this to be able to live, the products produced in our body and the scale of the body, pH scale of the body, should be around the neutral range. So everything has to be converted to the neutral area in order for the body to be able to live. As we have said before, pH refers to potential or power of hydrogen. Expresses hydrogen ion concentration in water solutions. Water ionizes to a limited extent to form equal amounts of hydrogen ions and hydroxyl ions, OH negative ions. Pure water is neutral. The hydrogen ions are equal to the hydroxyl ions and the pH is 7. When we move toward the acidic side, the hydrogen ions are more than the hydroxyl ions and the pH is less than 7. And if we move towards the basic side, the hydrogen ions are less than the hydroxyl ions and the pH is greater than 7. The pH range compatible with life is between 6.8 and 8. However, practically the pH value of the blood has to be between 735 to 745. pH equaling 4 is more acidic than pH equaling 6. pH equaling 4 has 10 times more free hydrogen concentration than pH equaling 5, and 100 times more free hydrogen concentration than pH equaling 6. So if we look at here, the normal range is around 7, and it's between 735 and 745. So this is the normal range of the blood. If it moves towards acidosis, the base limit that is compatible with survival is 6.8, and because venous blood carries more carbon dioxide, it is a little bit acidic when compared to the arterial blood, which carries less carbon dioxide and more oxygen. And on the alkalosis or the basic side, the number that is compatible with life maximum is 8. So life stays around 735 and 745, maximum to 750, and it's not compatible beyond 6.8 on the acidic side and beyond 8.0 on the basic side. An abnormality in one or more of the pH control mechanisms can cause one of two major disturbances in acid-base balance. The first one is the acidosis, and the second one is the alkalosis. An acidosis is a condition in which the blood has too much acid or too little base, frequently resulting in a decrease in blood pH. That is to say, it goes below 7. And alkalosis is a condition in which the blood has too much base or too little acid, occasionally resulting in an increase in blood pH. That is to say, going over 7. So acidosis and alkalosis are not diseases, but rather they are the results of a wide variety of disorders. The presence of acidosis or alkalosis provides an important clue to physicians that a serious metabolic problem is existing within the body, which is causing either acidosis or alkalosis beyond the limits of the compensation mechanisms that we have talked about. So pH changes have dramatic effects on normal cell function. First, changes in excitability of nerve and muscle cells occurs. Second, influences enzyme activity. And third, influences potassium levels. Well, when the pH decreases, it becomes more acidic, that is to say, it depresses the central nervous system, and it can lead to loss of consciousness. That is to say, when the body goes towards the acidic side, we start to lose our conscience. When the pH increases, it becomes more basic. It can cause overexcitability, and you start to have tingling sensations, nervousness, and muscle twitches. pH increases or decreases can alter the shape of the enzyme, rendering it to non-functional. Changes in enzyme structure can result in accelerated or depressed metabolic actions within the cell. So if you look at this figure here, if we have an enzyme like this, which is specific to bind with a specific substrate, and as the body shifts to alkalosis, severe alkalosis or severe acidosis, the enzyme will change its shape, and it will not be able to fit with the substrate, and the enzymatic actions will be halted or altered. So when reabsorbing sodium from the filtrate of the renal tubules, potassium or hydrogen is secreted. They're exchanged. Normally, potassium is secreted in much greater amounts than hydrogen. And if hydrogen concentrations are high, as it is in the state of acidosis, hydrogen is secreted in greater amounts. This leaves less potassium than usually excreted. The resultant potassium retention can affect cardiac function and other systems. So that is to say, if the body becomes more acidic, in other words, if it becomes loaded more with hydrogen ions, then the excretion of potassium is changed to the benefit of hydrogen ion to overcome the acidotic state. But this time, because potassium is not excreted through the kidneys, it starts to accumulate in the body, and the potassium levels will start to increase, and increased potassium levels will deteriorate, will be detrimental for the cardiac functions and other systems. And normal values of bicarbonate in arterial blood, when we look at the normal values, the pH is around 7.4. It's between 735 and 745. The partial carbon dioxide pressure in the blood is 40 millimeter mercury, and bicarbonate levels are 24 milliequivalents per liter. So mechanisms protect the body against life-threatening changes in hydrogen ion concentration by buffering systems in body fluids, by respiratory responses, by renal responses, and intracellular shifts of ions. So in order to keep these three parameters constant, these compensatory mechanisms are always working very hard to keep the balance. If we look at the buffer systems, though, there are the respiratory responses, renal responses, and intracellular shifts of ions. And buffering systems provide an immediate response to fluctuations in pH. The immediate response is done via phosphate, proteins, and bicarbonate buffer system. A buffer is a combination of chemicals in solution that resists any significant change in pH. It is able to bind or release free hydrogen ions. Chemical buffers are able to react immediately, within milliseconds. Chemical buffers are the first line of defense for the body for fluctuations in pH. The protein buffer systems behave as a buffer in both plasma and cells. Hemoglobin, which is found in the red blood cells, is by far the most important protein buffer. Proteins are excellent buffers because they contain both acid and base groups that can give up or take up hydrogen ions. Proteins are extremely abundant in the cell. The more limited number of proteins in the plasma reinforce the bicarbonate system in the extracellular fluid. The hemoglobin buffers hydrogen ion from metabolically produced carbon dioxide in the plasma only. As hemoglobin releases oxygen, it gains a great affinity for hydrogen, and hydrogen generated at the tissue level from the dissociation of bicarbonate carbonic acid produced by the addition of carbon dioxide. Bound hydrogen to hemoglobin does not contribute to the acidity of blood. So venous blood is only slightly more acidic than arterial blood because of the tremendous buffering capacity of the hemoglobin. Had it not been for the buffering capacity of the hemoglobin, we would have a normal pH on our arterial side and an extremely acidic blood on the venous side. So hemoglobin is compensating the acidity coming from the metabolism of the cells as hydrogen ions and carbon dioxide. Even in spite of the large volume of hydrogen ion generating carbon dioxide are carried in venous blood. Proteins can act as a buffer for both acids and bases. Protein buffer system works instantaneously, making it the most powerful in the body. 75% of the body's buffer capacity is controlled by proteins. Bicarbonate and phosphate buffer systems require several hours to be effective.